Hi, welcome all. This is the PGTai webinar. So today is the PGTA via the revolutionary platform PGT-A. So I'm David Chrimes. I'm the Director of Business Development in Cooper Fertility and Genomic Solutions, specifically the genomics part of the business. I'm a molecular biologist and geneticist by training. I'm not an embryologist. I want to make that very clear. So I'll be coming at this from a pure molecular biology and genetics background. So please bear that in mind, but I will do my best to answer any questions, as I say, to any aspect of this. And I've been working in the genetics testing of embryos for over 12 years now, and have seen the field advance a lot, including this technology. So certainly I've seen quite a lot of changes. So let's progress with this webinar. So just a quick introduction to CooperSurgical and Cooper Genomics. So we have over 27 years experience within the company of testing embryos for disease and aneuploidy. So we have the pioneers and founders, including Santi Monet and Marcus Hughes, for both PGS and what was PGD or PGTM as it is today. We estimated after today's calculation about 700,000 procedures of embryos and analysis. We have recently been reaccredited, and by that I mean had our inspection back end of last year for a new lab in London to ISO 15189, which includes the whole PGT testing suite. So that's great. We have a nice new lab to do all the testing in. We also have within house and genetics counselling help, which can be advised to the clinic, but we hope we're the driving force behind the advancement in PGT. So today I'll be talking about PGT-A, or pre-implantation genetic testing for aneuploidy. So just to make sure we all know what that is, PGT is now the generic term for pre-implantation genetic testing in embryos, and we have A for aneuploidy, M for monogenetic disease or single gene disorders, and SR for the structural rearrangements. So both PGT-M and SR are the diagnostic embryo testing, previously known as PGT-M. We won't really be covering those today in much detail. We will touch on PGT-SR. PGT-A in its previous has been previously known as PGS, pre-implantation genetic screening. It's also been known as CCS, comprehensive chromosomal screening. But it's a screen for all 24 chromosomes within the embryo. It's screening for abnormalities, both full copy number changes and also partial chromosome changes. It is an option for all IVF patients, and by that I mean we're not necessarily saying that every IVF patient should have PGT-A, merely that there isn't any real exclusion criteria from a patient which may have PGT-A, unlike PGT-M and SR, where you need a medical condition to really have those testing. And because of that, it does not require any kind of personalized test prep. So as I say, it's available to all patients. But ultimately, it's there to improve the IVF success rates for patients and clinics. So this is the PGT-A process. So the IVF is carried out by the IVF clinic. Now, I just want to reiterate, at least through Cooper Genomics, that you do not need to do ICSI just because the patient is having PGT-A. We have comprehensively validated our technology that it cannot amplify DNA from any contaminating sperm. So really, although initially PGT-A or PGS as it was recommended ICSI, there is really no need if you have a patient where ICSI is not appropriate. They're still open for PGT-A. A trained and highly skilled embryologist would then carefully remove a small sample, 5 cells roughly, from the embryo of a blastocyst. That's generally day 5, 6, or 7. After the biopsy has been taken, then the embryos are vitrified, and this is important to state because, at least for Cooper Genomics, all embryo, this is based on frozen embryo transfer. So the time it takes to generate the results does not facilitate a fresh embryo transfer. So you send the biopsy samples to our PGT lab in London, where we perform the testing, and then we release the results to the IVF centre. It then becomes the responsibility of the clinic and patient discussion to decide which embryo to transfer, and you would typically transfer a euploid, a normal embryo first. Any additional euploids or perhaps even mosaics can be frozen for future transfers if available. So PGT-A is all around the chromosomal health of embryos. Now, what we mean by that is an embryo should have the correct number of chromosomes, or the normal number, because we know these have the highest likelihood of a successful pregnancy. Now, this information actually came from natural conceptions where one of the leading causes of miscarriage is actually aneuploidy in the embryo. So when people started looking at product of conception testing, so those are the products of the miscarriage, what they found is a very high frequency of those miscarriages were the direct result of a chromosomal abnormality. And so the theory of PGT-A, or PGS as it was then, was if you could find the chromosomes which are the embryos, sorry, which are chromosomally normal, then you decrease the chances of that embryo going on to form a miscarriage. So here we just have our normal embryo with, if it was female, it would be XX at the bottom here, or if it was male, XY. But all the other chromosomes, 1 to 22, are what are referred to as the autosomal chromosomes. They're shared between both male and females. Here we have an aneuploid embryo, and here we've highlighted trisomy 10, an embryo with 3 copies of chromosome 10. So this is an incorrect number of chromosomes. We know that aneuploidy is found in embryos of IVF. We also know that aneuploidy increases with age. And again, this isn't just an IVF issue. This is a natural fertility issue. It's one of the reasons why you see an increasing chance of Down syndrome, trisomy 21, as maternal age increases. But what we do know is that a trisomy may, or even a monosomy, may result in a failed implantation. So the embryo is transferred, nothing happens. It does implant and you have a miscarriage, and these can be either very early in the pregnancy or in some cases quite late into the pregnancy. And depending on the type of abnormality, that can actually be a genetic disease. The reason that we've highlighted chromosome 10 here is specifically because there has never been a reported live birth globally with a trisomy 10. So basically, if we find an embryo with a trisomy 10, then we're very certain that that embryo could not go on and form a healthy live birth or even a pregnancy. So what is the point in transferring that embryo? And again, that is the point of PGT-A. Now, what we have come to learn over the years as technology has improved and more embryos have been biopsied is we can have what's known as mosaicism in the embryo. So mosaicism is very simply an embryo where there's a mixture of cells, at least 2 different cell lines, and the blue here are the aneuploid cells and the green are the euploid cells. And if you have a mixture of these cells, what we're now seeing is that these implant less. So if it's a mosaic embryo detected by PGT-A, they implant less, they miscarry more, but they can sometimes lead to live birth. So what this tells us, and from all the literature and the studies of mosaic embryo transfer shows us, is that correct identification of euploid, mosaic, or aneuploid embryos is essential to making informed decisions on which embryos to transfer and embryo prioritization. So again, PGT-A is there to prioritize embryo transfer to maximize the success of each embryo transfer. It is not there just to decide a yes or no answer. It's a prioritization tool. So at Cooper, we have followed the PGTIS guidelines for how to report embryos and split embryos into 4 categories. Euploid, which are your normal, and these have the highest chance of success. What you then have are low-level mosaic. So these are 20 to 40% abnormal cells. So these are lower chance of implantation success, but they're still moderate. They should certainly be considered. What we have then is high-level mosaic. So these are 40 to 80% abnormal cells, and these have a much reduced chance of implanting and a much increased chance of miscarriage. But again, occasionally they can go on and form a healthy live birth. And then you have the truly aneuploid, where every cell is abnormal, and that is very, very low, very unlikely to actually result in a healthy live birth. So if we look at the PGTISI, PGT-ASM platform, so it was a groundbreaking platform. It harnesses the power of AI, and really we're using big data and machine learning to ensure we have the highest sensitivity and specificity during PGT-A testing of embryos. We could do this because we've been doing PGT-A for many years. We are now collecting outcome data. Well, we've always been collecting outcome data, but we've been collecting outcome data on NGS technology PGT-A. And we have reanalyzed over 1,000 embryo data, which resulted in live birth and sustained pregnancies. So we had the amplified DNA from embryos that were previously reported euploid. They have been transferred and gone on to perform a sustained pregnancy or a healthy live birth. So we knew whatever we saw in that embryo profile had not had a significant impact on that embryo's ability to go on and form a healthy live birth. Then we sequenced over 10,000 embryos and analyzed them. And by that, I mean we compared the data to the algorithm, to a visual inspection, and saw if the algorithm made sense. So we went through 10,000 embryos one by one to check that the algorithm had called the embryos in a way that made sense. And this added a statistical robustness and a user independence to analysis. So by removing subjectivity, we know that whoever has analyzed that embryo in the lab, or their series of people, they're not having any input into how that embryo is scored. So you have complete user independence of reporting. And because most of it is automated, we avoid many of the human errors of data transcription. We all know if somebody has to keep copying and handwriting text, eventually mistakes get made. The more you can automate, the more that you can remove those human transcription errors. So prior to that, and still for many companies out there today, most of the PGT-A is a subjective data interpretation. So even if they do NGS, which many do, what that is, that NGS is converted into a visual image that users can read, and they make a call as to whether that looks normal or abnormal, and then that information is fed to the clinician to make a transfer decision. Now, we all know from embryology grading and various other studies in IVF that you give the same data to 3 different people and they won't always agree as to what that answer is. And this is your subjectivity in calling. So what we did is we still do the NGS, but we use the mathematical algorithms and machine learning to drive that decision. Machines don't get bored, they don't get tired, they're not subjective, they just follow rules. And this means that we have a very consistent calling criteria, which means that when you transfer that embryo based on a call, you're incredibly confident that there's that call is correct and accurate. So what has this resulted in? Well, we've reported more euploid embryos as a percentage of total embryos tested. Another key metric is we've increased the number of patients with at least one reported euploid embryo, so they may have only had mosaics and aneuploids before. And what that resulted in is an extra 1,300 euploid embryos being reported between November 2018 and May 2019. So if we're claiming that we're reporting more euploids, where have they come from? And what we've actually seen is a big decrease in the relative amount of embryos that previously were being labeled as mosaic, particularly low-level mosaics, and they are now being categorically scored as euploid embryos. So the relative increase in euploids is about 7.7%. We have also decreased the number of aneuploid embryos, with some of them moving into a high-level mosaic. Perhaps we were being overly cautious in calling some as aneuploid, which were actually high-level mosaic. And the good news is that these embryos were distributed across every age range. So it wasn't like this technology was only benefiting a certain subset of patients. Now, in the UK and much of the international market, the average age of patients undergoing IVF is above 35. And certainly in the UK, the average age, over 60% of patients are 35 and above. And we typically see IVF average age of PGT-A patients being around in this 38 to 40 age range. And so here we saw a 4.2% increase in the number of embryos that we were confidently reporting as euploid. And these embryos were spread across the whole population. So it wasn't like a subset within this population were getting euploids. The good news was that actually there's about 1 in 25 couples that previously only had a mosaic or aneuploids were now having a euploid embryo for the easy transfer decisions. So it's really helping the clinics make a good transfer decision. We weren't happy with that. We felt there was more that we could be doing. So we launched PGTi 2, and all samples processed after the 30th of March in the UK lab will be run on this technology by default. But it's got the same underlying backbone of these 1,000 live births and sustained pregnancy outcomes, but we now sequence 10 times to the depth that we used to do. So for every single sequencing read that we used to do, we now have 10 of them. We also do paired end sequencing, so that means you read from both ends of the DNA fragment. That means you have better alignment and, again, more depth of coverage. And that depth of coverage has allowed us to get SNP coverage. Now, SNPs are single nucleotide polymorphisms, and we'll come to those in just a second. But having them in across the whole of the genome content is a world first for PGT-A, and is actually the standard of care in postnatal and prenatal testing. So we've basically brought the embryo screening technology to a level which is very similar to that now used in postnatal and prenatal testing of children with genetic abnormalities or abnormal scans. And these two independent methods for aneuploidy detection gives us the increased accuracy and sensitivity for calling embryos. So what that has allowed us to do is make the most robust assessment of embryos in the market. We can now detect all forms of ploidy. So we've always been able to detect male triploidy, so that's where it's XXY or XYY. Now, in the plus version, which requires the submission of both parents' DNA through a cheek swab, we would actually be able to detect and confidently detect both female haploids, so that's just a single copy of the female genome, or full female triploidy, so the embryos which are fully triploid female, previously undetectable by NGS, and for many firms still undetectable. This allows you to do the 2PN validation, so anything that comes back as either not triploid must be 2PN. And what we can also offer for patients, if they're prepared to submit cheek brush swabs, is the parent of origin of abnormality. And what I mean by that is if the embryo comes back as an aneuploid and it's got, say, a trisomy chromosome 9, has that extra chromosome come from mum or has it come from dad? For most people, that won't matter. It doesn't change things. But if they have all their embryos from a cycle coming back as aneuploid, and you wouldn't expect that for a person of that age, then you can start seeing if there's a sperm issue or an egg issue, and which partner is potentially driving that aneuploidy rate, which may make a difference to treatment or donor decisions. So we can do all this because, as I said, we're detecting SNPs, which is a common form of genetic variation. So a SNP is a single nucleotide polymorphism, and a single nucleotide is your basic building block of DNA. So all of us contain approximately 3 billion DNA base pairs. So we have potentially 3 billion sites for these differences between me and the person walking next to me down the street. And they result from natural variation in human reproduction and evolution. So most of these SNPs have no effect on health or development. They may be something as simple as, say, hair color, that gives you different tones of hair color. So they may have a phenotype, but they're not disease causing. Others are completely hidden. Of course, there are SNPs which do cause disease, which we shouldn't ignore, and these are generically called mutations, and these are what may cause cystic fibrosis and other single gene disorders, but they're still SNPs, but they're generally referred to as mutations. But SNP patterns provide a secondary aneuploidy assessment. So it's estimated that all of us have approximately 5 million of these SNPs in their genome. They act as biological markers, and you inherit it, the vast majority, from your parents. And this, if we think of testing, this has been going on for years, looking at inheritance patterns and paternity testing. There's all sorts of inheritance disease linkage testing where you're looking at these unique markers and tracing them through generations, genealogy testing, 23andMe, all these firms looking at your heritage. What they can also do, and what we use them for, is assessing potentially whether that monosomy or trisomy has come from mum or dad, whether you've got just a copy from mum or one copy from dad, or in this case 2 copies from mum plus a copy from dad. It is done from the same biopsy, so we get the biopsy cells into the laboratory, we do the whole genome amplification, and from the same sequencing run, so this is a single sequencing run, we get 2 different forms of information. We get your gold standard copy number chromosomal counting, which is what PGT-A testing has been based on for many years. But as a completely independent measure, we also get this SNP call. And this shows us how many alleles are in each region of the genome. And what this allows us to do is remove a lot of the artifacts that you may have seen in previous plots. It gives us best-in-class mosaic calling, so by removing these artifacts, we're much more confident if it is truly mosaic or not. But what it really, really allows us to do, and this feeds into PGT-SR, the structural rearrangements, is be able to very accurately call down to 5 megabase resolution. So if you're a translocation carrier, these are quite often small changes at the ends of chromosomes, and by having this increased resolution, we can more accurately detect the chromosomal imbalances in embryos and inherited patterns when it comes to PGT-SR cases. So although this is mainly talking about PGT-A today, this secondary confirmation really is important when it comes to PGT-SR cases as well. So what we have is two test options. So your basic PGT-A, your PGT-AI2, as after the 30th of March, will have this copy number for mosaicism through both the CMV and the SNP calls, and there's no change in your pricing for that test. What we will also be launching is what we're calling PGT-AI2 Plus. And what this gives you is your parental of origin of aneuploidy, so working out if the aneuploidy is from mum or dad, whether you get your female triploidy information. So if we have parental cheek swabs, we can tell you whether that embryo is truly haploid, diploid, as it should be, or triploid, including female triploidy. To get these samples, we can ideally send the cheek swabs to the clinic, although in some cases we could do it to the patient. But it's also available as a retrospective test. Sorry, there is a typo in there, but there is, if you have samples that have been already analysed on PGT-AI2, so anything analysed after the 30th of March, then we will be able to retrospectively get cheek swab samples and go back and look at those samples for that missing information if it's deemed important. And the reason that this could be important is there are studies out there and assumptions are made, but we know that there is variability among aneuploidy. So generally speaking, meiotic whole chromosome aneuploidy is being determined to be maternally derived in 90% of patients. The segmental changes is generally considered to be paternal. However, as we know, each patient is different and each story is different. So by testing for this origin of aneuploidy, you may be able to make more confident donor monogamy decisions, whether to go for a sperm donor or an egg donor earlier in the process, or potentially just providing the patient a peace of mind, and that may be all they're looking for. So by understanding where this aneuploidy is coming from, hopefully more confident decisions can be made on future treatment. So just as a summary of where we are for PGT-AI and PGT-AI2, we have taken what was already the best-in-class technology and taken it to the next level. So the basic test, if that's the right term, the standard test is going to be providing these two independent assessments of ploidy, so both the CMV and the SNPs. And this is derived by being able to generate more data and the deeper analysis. If the patients are willing and the clinic wishes for it, then with the addition of the parental cheek swabs, then we can do these extra 3 tests for parent of origin testing for meiotic changes only, the triploidy, and the 2PN verification. So PGT-AI will continue to evolve, it will continue to improve, and this is where we are today. But the whole reason that we're innovating in PGT-AI is to try and get the best clinical outcome and the highest clinical impact for your patients. So we want to ensure that you have the maximum number of euploid embryos available for transfer. However, we also want to be sure if we're calling them a euploid that they are, as far as we can tell, truly euploid and not a mosaic. Equally, if we say that they are mosaic, we want to be sure they're mosaic and not an aneuploid or a euploid. We want to decrease miscarriage risks. So if we can identify any samples with either extra forms of triploidy or aneuploidy, or indeed low-level mosaicism that was previously missed. Anything that we can do to decrease miscarriage risk is a good thing for patients, it's a good thing for clinicians. Miscarriage is one of the most traumatic problems with fertility, and in fact a recent study has shown miscarriage to potentially be as devastating as a cancer diagnosis for some patients. So absolutely should not be underestimated, and if we can give the best possible embryo grading, then we should see a positive impact on pregnancy rates per embryo transfer. It's all very well us innovating technology, great answers, but we are just part of the chain. There are a whole aspect that goes into patient care from the clinician, embryologist, doctors, all sorts of things. So what we want to do is ensure that you know that we are there to support you and we have quality assurance. So from a support perspective, as mentioned earlier, we do have in-house genetics counsellors that can be contacted and can provide guidance to both clinicians and patients in terms of training and common questions and how we can answer you. We are ISO accredited as we need to be for these tests. We have a very strong commitment to the quality of what we're doing. And what we also do is looking for partnerships and collaboration. So we have our own ideas as to what will advance the field in IVF, but we don't live, breathe, and work in an IVF clinic every day. So we're open to any partnership and collaboration ideas which could improve the outcome of technology. Yes, this is a genomics talk, but it doesn't have to necessarily be genomics. I'm sure you're aware CooperSurgical have bought many IVF firms over the years. We have catheters, media, all sorts of things. So think about ways that perhaps a research partnership may be open. And then when we do research stuff, we have over 700 scientific abstracts and publications. We're looking to innovate in the field, as I say, and we see this again as gaining trust in the market and showing our commitment to the market. So I'm just going to finish the webinar with this slide talking about that we are supplying the revolutionary PGT-A platform for PGT-A in the international market, and as of March the 30th, all samples will get this new test, an improved test. Please feel free to contact us at global support at CooperGenomics.com, or you can contact me personally on my email that's on the screen now, david.chrimes at CooperGenomics.com. We will happily take any questions. With that, I thank you for your time and I look forward to talking to you soon. Thank you.